Hey guys, welcome to Metabolism and Enzymes 2.5.2 or part 2 of our Metabolism and Enzymes lecture. Okay, so let's look at this picture at the very beginning, and what we're seeing are two different models. The model on the right is a cartoon of an enzyme, which is a protein, and the molecules it is operating on, the substrate molecule. And that's our little cartoon model, and we'll be seeing lots of these. Off on the left-hand side is more of a what we call a space-filling molecular model of an enzyme, and this is kind of what it looks like in the real world. And what I want you to notice is look at the size of the enzyme. In this case, it's a model of the enzyme catalase, which we all have in our bodies. And its function is to break down hydrogen peroxide, otherwise known as H2O2. As you can see, that is the substrate molecule. The substrate molecule, H2O2, is going to fit into the active site of this catalase enzyme, and catalase will break it down into harmless products of water and oxygen gas. And again, hydrogen peroxide is produced in our bodies during the breakdown of proteins, and our bodies need to get rid of it. So this is the picture I want. And so what's going on here? Well, this is a reaction. This would be an example of a catabolic or hydrolytic reaction. This is hydrolysis. We start off with our reactants, and let's say it's a sucrose molecule. Okay, that's table sugar. Our reactants, as you can see, have a lot of energy. That's that free energy. They're up high there, about the middle of the graph. And this is going to be a hydrolysis reaction. When we break that sucrose molecule up into two separate monosaccharides, we're going to end up with the products that have less energy. Where did the energy go? It was released into the system, in this case, our bodies, and it was either used to drive another reaction or it was used to heat up our bodies. Okay, because energy coming into the system, it can't disappear, it just gets transformed into another form. In this case, it gets transformed from the energy in the bonds to heat energy, and that's what happens. And what we see here, we see two graphs. We see the red one and we see the black one. Well, the black one is the reaction without the enzyme. And what do you notice about this? The black one goes much higher up than the red one does. Okay, that means it's much harder for this reaction to occur without an enzyme. It's difficult. We call that the energy of activation, or E of A. So why do molecules have an energy of activation? Why is this even necessary? It's necessary because if there was not an energy of activation, nothing would ever stay together. It would simply just fall apart immediately. Two molecules would come together and they'd immediately break down because that's the more stable condition. When you put two molecules together, it's actually kind of unstable. They want to break apart. Okay, they want to go to equilibrium. So enzymes can only catalyze reactions that would actually happen in the first place. And again, looking back at this diagram, the energy of activation without an enzyme, as you can see, is much higher than it is with an enzyme. With an enzyme, it's much lower, and the reaction is more likely to occur with an enzyme than without. And this can be a tremendous difference. I already mentioned in class that the breakdown of hydrogen peroxide from H2O2 to water and oxygen gas, that can happen on its own, albeit very slowly. One single catalase molecule, one single catalase enzyme molecule can undergo 40 million reactions per second. So it speeds up this process tremendously. Okay, so here is the way an enzyme works. It's a cycle, and that's an important thing. An enzyme is not going to be used up nor altered during its reaction. Again, I will say that once again. Enzymes are neither used up or altered during their reactions. And so let's follow what's happening here. Here we have a substrate molecule, sucrose. the enzyme sucrase. Notice OSE for sugars, ASE for enzymes. So sucrase breaks down sucrose. Sucrose binds to the active site. That's where the action's all going to happen. Okay, so once the sucrose and the sucrase bind together, that's known as the enzyme substrate complex. And since this is a hydrolysis reaction, water has to come in. The water comes in, the enzyme basically splits the water, adds the OH to the glucose molecule, adds the hydrogen to the fructose molecule, spits them back out, and then the enzyme sucrase is ready to start over again. And so we start with our substrate sucrose, we end up with our products of glucose and fructose, and we end back up with our enzyme ready to go again and ready to do another cycle. So once again, the enzyme is neither used up nor destroyed during this reaction. And then the enzyme drops off very steeply after that optimal point. That's because the enzyme begins to denature and fall apart. Um, Before the optimal point, it's just that the molecules are moving slower and they just don't bounce into each other as often. So the cold temperatures don't really destroy the shape of the enzyme, they just slow everything down. It's the high temperatures that can destroy or denature an enzyme. And that's what this means. Denature of an enzyme. That's when a protein unfolds. And which bonds do you think are going to break first? Strong bonds like covalent bonds or weak bonds like hydrogen bonds? You got it. Those hydrogen bonds are going to break and that protein is going to unfold. It's going to denature. And if we go back to the normal conditions, the protein may renature back into its normal shape. It's a possibility. If the temperature gets too high, that won't happen. And also one thing is that as we will model or have modeled in class already, other things can stop a chemical reaction, an enzyme catalyzed reaction. We call these inhibitors. And we have two types of inhibitors. One is a competitive inhibitor, which competes with the substrate molecule because it actually goes into the active site. And the other one's called a non-competitive inhibitor or an allosteric activator or allosteric inhibitor. And it doesn't bind in the active site, but it binds somewhere else on the enzyme, and it causes the active site to change shape. And once you change the shape of the active site, the substrate molecule will no longer fit there because it's a very relatively precise fit. And that's going to basically shut down that biochemical pathway catalyzed by that particular enzyme. So why do enzymes do this? Why is there an inhibitor molecule? Well, it helps to save energy. Why produce something if you don't need it? Okay, so here's an example of what we call a negative feedback inhibition. We start off with our initial substrate, which is threonine. It binds into the enzyme 1, and over a series of 5 reactions, it will turn into the product isoleucine. Well, if there's a whole bunch of isoleucine in the cell, there's no need to make any more. So if there's extra isoleucine around, it will actually bind to an allosteric site. Allo means other, steric means site. So someplace other than the active site, and it's going to cause a shape change in the enzyme so it no longer can bind to threonine. As as isoleucine gets used up, then it will be used up in that allosteric site, and that will cause the enzyme to shift back into its normal shape. So threonine can now bind back in there again, and we can start the pathway all over. And again, it's all a means by which to save energy. And remember, those organisms that save energy in doing these little things are going to have more energy left over for other things like taking care of young, producing more babies, and so on and so on. So it's sort of a process of natural selection that leads to this. Okay, so we'll talk more about this later. This is called negative feedback inhibition. It kind of works like your furnace. You put your furnace on at, let's say, 68 degrees. When it drops below 68 degrees, the temperature turns on, and your furnace heats up the air until you get to 68 degrees, and the thermostat figures that out or senses that. It turns the thermostat off, and it keeps the thermostat off until the temperature drops below 68 degrees again, and then it comes back on. That's also negative feedback inhibition. So anyways, hope you enjoyed this relatively short lecture at 10 minutes, and we'll see you in class.